So today we're going to talk about the genetic code. But before we talk about that, I just want to review what we covered in class yesterday. So yesterday we talked a lot about proteins and how proteins do so much in our body. Therefore, our genetic information in DNA has to hold the instructions for your bodies to make proteins. So that brings us to the central dogma of biology that we talked about yesterday. And that is that DNA codes for RNA, which codes for proteins. Does that sound familiar from yesterday? Okay. And then at the end of class yesterday, we did that coding activity together as a class. And that activity was to allow us to understand what are some coding rules we have to follow, just to kind of help set us up for today. So today we're going to pick up with coding, and we're going to talk about our genetic code. So once the pattern of information flow was determined in our cells, that means that our info goes from DNA to RNA to protein. The next step is to figure out how does that actually happen. Okay? So how do we actually take the base sequence of mRNA and how does it actually code for proteins? And so to help us understand that, we need to figure out the rules of our genetic code. Okay? So what is the relationship between a sequence of nucleotide bases in DNA or RNA and the corresponding sequence of amino acids in proteins? All right, so the first thing we have to determine is how long is a word in our genetic code? So remember yesterday, we did that coding activity with like the cats and the mice and the dairy products. How could you tell one word from a different word in yesterday's code?
Has 2 C's and 2 M's.
Okay, there is like an order that there are 2 C's. So how does your body read your code and separate like one word from the next word? Okay. So a guy named George Gamow predicted each word in a genetic code just contains 3 bases. So every 3 bases makes a new word. He did this just using like simple math and just a thought process. I'll show you a picture on my next slide of his thought process. But the way he kind of thought about it was there's 20 amino acids. So what is the least number of base combinations that could code for up to 20 amino acids? And so the simplest answer is 3 bases can make a combination of greater than 20. So if we have 4 letters and 3 of them are going to combine, it's just 4 cubed, or 4 times 4 times 4, which equals 64. So if a 3-base code gives us 64 different combinations, that's more than enough to code for the 20 amino acids. And so that 3-base code is known as a triplet code. So here's just a picture showing you George Gamow's thought process. Okay. So we know that there's 4 bases in RNA. There's U, C, A, and G. We also know that nature works. The simplest form, things in nature behave the simplest way possible. Okay. So if we just start that 1 base codes for an amino acid, there's 4 bases, so 4 times 1 is just 4. 4 codes is not enough to code for 20 amino acids. Okay. So we know it has to be more than just 1 letter. So then we move on to 2 letters. Okay. So what if we take these 4 different letters, and what if it's just codes of 2? Okay. So to figure that out, all the combos are just 4 times 4, which equals 16. Okay. So 16 amino acids is not enough to get to the actual 20 that exist. Okay. So then he moved on to 3. So what if we took those 4 letters and grouped them into combinations of 3s? So to figure out those combinations, it's just 4 times 4 times 4, which is 64. So 64 gives us more than enough combinations to code for all 20 amino acids. And since nature behaves in the simplest way, there's no need to keep going. We would know that 3 letters makes up the code. So then how long is a word in the genetic code? It's 3 letters long. Okay, so you could think of every 3 letters would be a new word. And instead of calling it a word, it's really going to be called a codon. Okay, so get comfortable with the word codon. So that means when we look at 3 letters, that's going to make up like a word or a codon. The next 3 letters make up the second word or the codon. And then, the next 3 make up the following word or codon. So we read our DNA or RNA sequences in groups of 3. All right, so in addition to knowing how many letters form a word or a codon, we also know how to start, how to stop. Okay. So there is such thing called a start codon. Remember yesterday when we looked at those codes, what did every single code start with? Run. Okay. So this time in your actual RNA, it's going to start with AUG. So whenever your ribosome sees AUG, we know it's time to start making proteins. Okay. So that's the code that says start. We also need to know the code when it's time to be finished. So yesterday in our activity, what was the word that let you know you were finished? Done. Okay, so in your RNA, instead of done, it's either UGA, UAA, or UAG. So whenever we're reading any of those 3, that means it's time to stop. The protein is finished. So then let's do a little bit of math. So if we know that there were 64 total combinations, 1 is for a start codon, 3 code for stop codon, so that leaves 60 left. So those remaining 60 combinations are going to make up the 20 amino acids. So here's a chart that shows you how do all the different codons and which amino acid corresponds. So let me show you how to actually use this chart. So if you take a look at this first vertical column, this is the first letter in the codon. This row up here corresponds to the second letter in the codon. And then this column over here is the third letter. So for example, let's say we just want to find the codon, let's say CCC. Those are the 3 letters we're looking for. So here's the letter C. So first letter is C. Then I'm going to go over to the second letter is C. And then I have to line it up over here. Okay, so I get CCC. And this is going to code for the amino acid PRO. Let's do another one. Let's find AGA. Let's find AGA. So again, we use the first letter A, so I'm in this row. Then I go all the way over to G, okay, from this second column. And then I line up the third A right here. So AGA codes for the amino acid ARG. Okay, you find GAC. So you're looking for what amino acid corresponds to GAC? What are the 3 letters? Good. Okay, so G is here, A, and then did I say GAC?
Yeah.
Okay, so C's here, it lines up to ASP. So do you think you could use this chart on your own?
Yeah.
Good. All right, so now that we know our code, there's a couple properties that correspond to that code. So first of all, it's redundant. So that means all of the amino acids except 2 are encoded by more than 1 codon. So let me explain what I mean. If you look at your chart, just look at like CUU. Okay. Look at CUU on this chart. It codes for an amino acid with this abbreviation LEU, which is leucine. But do you see how there's 4 different codons that all code for leucine? Okay, so that's what I mean by redundant. Multiple codons can equal the same amino acid. It is unambiguous, which means 1 codon never codes for more than 1 amino acid. So if we're still using CUU as the example, CUU always codes for leucine. It's never going to code for anything else. It's nearly universal, so there's just a few exceptions, but for the most part, all codons specify the same amino acids in all organisms. So, for example, I'm going to use CUU. That's my example. CUU codes for leucine in my body, but it also codes for leucine in like my dog's body, and it's going to code for leucine in my cat's body. Okay, so CUU is always going to equal leucine. And then finally, it's conservative. So the same, or the first two bases are usually the same when multiple codons code for the same amino acid. So go back to the box where CUU is located. All four of those codons code for leucine. And do you see how all four start with CU?
Yeah.
Okay, that's what I mean by it's conservative. Okay, so now that we know the rules of our genetic code, so how to distinguish our words, how to tell when to start, how to tell when to stop, now we're going to move on and talk about transcription and translation. Or how do we actually get from our DNA to mRNA to proteins? So our cells build proteins using the instructions found in our genome or our genetic info, our DNA. And we follow the central dogma of biology. That got cut off in my formatting, but this should be central dogma of biology. And this picture should look familiar from yesterday, yes? Okay, so DNA is going to code for mRNA through a process called transcription. And then mRNA codes for proteins through a process called translation. So today we're going to look in depth at transcription and translation. So transcription, it's just that first step where we take the genetic info stored in your DNA and we use it to make a new strand of mRNA. And so the enzyme responsible for creating that RNA strand is called RNA polymerase. So it reads the DNA and it makes that new strand of mRNA, which is also called the template strand. So think of this as a template that's going to be read to help produce a protein. All right, so here's a picture of transcription taking place. So this red strand at the bottom, this is the DNA. And then the yellow strand is going to be the complementary RNA. So I just want to go through this picture and show you how RNA is created using DNA. Okay. So DNA's bases are right here. So we have G, G, A, T, C, G. So when RNA polymerase comes through, it builds a strand of RNA that's complementary to the DNA. So DNA has the base of G. What letter is complementary to G? C. Okay, so the RNA has a C here. Here we have the letter G. Again, what letter is complementary to G? C. Okay. Next, we have the letter A. Now, usually if it was DNA, what letter is complementary to A?
T.
T. But there is no T in RNA. Instead, it's U. So you get a uracil here. Next is the letter T. What letter is complementary to T? Okay, so an A would go here. Then we have the letter C. What's complementary to C?
G.
Okay, then we get a G. And we have another C, so what's complementary?
G.
G. Okay, so RNA just comes through and it creates the complementary strand. And when RNA comes through, it moves in the same direction as DNA polymerase, and it moves in that 5' to 3' direction. All right, so now I added a slide. So this isn't in your notes packet, but find a space like on the side of your notes, or you can use the pink chart I just gave you. I want to practice converting this strand of DNA into mRNA. And before we do that, I just want to make like a little table off to the side. I'm just showing like what strand of RNA is complementary to DNA. Okay. So what are the four bases for DNA? What are those four letters?
A, T, C, G.
Okay, so we have A, T, C, and G. So then over here, I just want to write what's the complementary letter. So what's complementary to A? It would usually be T if it was DNA, but is there a T in RNA?
It's U.
Nope, so it's U. Okay, so we're going to put a U here. And then what letter is complementary to T?
A.
What about complementary to C?
G.
G, and then what about
C.
G? C. Good. Okay, so if you need to just put like this little chart on the side of your paper, feel free to. The tricky one is going from A to U rather than A to T. So you might need to write this down just to give you like a visual reminder. So now that we have this visual reminder, I want to go through and now I just want to write down our new strand of mRNA. So the first letter of DNA is A. So what's the complementary RNA base?
U.
Okay, so we're going to have a U. Then is a T. So what's complementary to T? A.
G.
Okay, then we have a G.
C.
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 U.
U.
G.
G.
A.
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 U.
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 C.
C.
G.
G.
A.
A. Good. Okay, do you feel okay with that? Okay. Could you do this on your own?
Yeah.
Okay, this is it. That's all transcription is. So all we do is polymerase reads DNA, and it just makes that complementary strand of RNA. Okay, so now I want to switch gears and jump to translation. So I deleted just a couple slides. So take a look at your notes packet. I'm skipping maybe like 2 sections a little bit. So go find where it says an introduction to translation. Okay. And I just took out that extra info. I took out what I thought wasn't necessary. Okay, so we're jumping down to an introduction to translation. Does everyone see where we're at on their paper? Okay, so once we code for an mRNA strand, the next step is translation. So we're going to read this mRNA, and we're going to use that to create amino acids, which are going to bond together to make that protein. And so this is going to happen in the ribosome. And the ribosome is going to be what is going to catalyze translation, and it's going to read that mRNA and actually make the protein. So here's just a picture of the cell, large picture. So this is our nucleus. Here's our strand of DNA. Okay. So RNA polymerase is going to read this, and it's going to make mRNA. mRNA is mobile, so it can move out of the nucleus and into the cytoplasm. And there, a ribosome is going to meet it in the cytoplasm. So then the ribosome, it's going to read this mRNA strand and produce a protein. So today we're going to look at like the more in-depth steps of how this actually happens. So we know in the ribosome, we have this strand of mRNA. And we know that 3 bases of mRNA are called a codon. And then we know which codons code for certain amino acids. So we're left with kind of this gap here. So Francis Crick proposed there has to be some sort of adapter molecule that can read the mRNA and somehow figure out which amino acid it codes for. And so subsequent studies showed that there is indeed this adapter molecule. It's called tRNA, which is transfer RNA. So transfer RNA is just this molecule. We say it's charged with an amino acid, and that's found at like the top of the tRNA. So it has one of those amino acids. At the bottom of the tRNA, it has something called an anticodon. So it has 3 RNA bases, and these are complementary to the codon found in mRNA. Okay. So if the mRNA has a U, the complementary letter is A. Complementary to C is G, and then complementary to A is U. Okay. So this anticodon is going to line up with the codon. And so that's how we know which amino acid is going to code for each codon in mRNA. So all of this happens in the ribosome. So I just want to talk about the structure and the function of the ribosome. Ribosomes are made up of 2 main things, so just protein and ribosomal RNA, which is rRNA. So if you take a look at this picture, this red-brown structure, it kind of looks like a poofy hamburger bun. Okay, if you use your imagination, can you see this would be like the top of your hamburger bun, this is the bottom. And then it opens up, so your mRNA strand goes in the middle, kind of like your hamburger patty would go. And then in the ribosome, do you see there's these 3 spaces here? Can you see there's like 3 shaded regions? Okay, so these are locations for 3 different tRNA molecules to come in here. So over the next few slides, we'll talk about what actually happens. So first, I just want to start with those 3 locations. Okay. So the first spot is called the A site. So this place in the ribosome, it's where we accept a new tRNA molecule. The official word is like amino acyl. It just means it's carrying the amino acid. That's more like biology jargon than we really need. So just think tRNA. Okay, so A site is for acceptor. So think where the tRNA enters. That middle site is called the P site. So this is where the amino acids are going to bond together. And the bond between them is called a peptide bond. As they grow, they're going to make something called a polypeptide chain. Okay, so P site correlates with peptide. And then the third and final site is called the E site. This is the spot for when the tRNA has already dropped off its amino acid, and then this is where it's going to exit the ribosome. So think E is for exit. So here's a picture of a ribosome. So here's the A site. So this is where the tRNA is going to enter. The P site is where we're going to form a peptide bond in between the amino acids. And then the E site is when the tRNA has already dropped off its amino acid and it's going to exit. If you read the order from right to left, it goes APE. What word does that make? What's APE? Ape, right? So remember ape. All right, so now let's write down what actually happens in those sites. Okay, so in that A site, the tRNA carries the amino acid, and its anticodon is going to match the mRNA codon. Then the next step, a peptide bond, is just going to form between the amino acid that's in site A to the amino acids that are already in site P. And then after that bond is formed, the entire ribosome is going to shift ahead 3 bases. And so then all 3 of those tRNAs, they're going to move down just 1 position. Okay. So then if there's a tRNA in the E site, it gets booted from the ribosome and it exits. So I do have a video. I'm going to play this at the very end, just when I use like my screen sharing software, it won't let me play YouTube. So we'll watch this in just a couple minutes. So if we continue on with translation, there's 3 phases. There's initiation, elongation, and then termination. All right, so that first stage is called initiation. What does initiate or initiation mean? Start, okay? So this is where we start to make a protein. So when the ribosome reads AUG and the mRNA, this is what is the initial, okay? So this tells your body to start making proteins. The second stage is called elongation. What does it mean to elongate? Good, stretch, right? You're going to get longer. So this is the stage where we actually build that long protein. Okay. So this is the stage where you have the tRNA at the P site. Okay. At the very beginning, the E and the A sites are empty. So then a second tRNA moves into the A site. And then a bond is going to form between the tRNAs and the P site and the A site. Okay. And then after this, we're going to see a shift. So then the ribosome is going to shift over, and then a new tRNA can move in, and we can keep building on this chain. So here's just a diagram that shows you what happens during elongation. I'm going to start in this picture, and then we'll work our way over. Okay.
The last one,
So we start with a tRNA in the P site. So then a new tRNA is going to move into the A site. The correct tRNA moves in because of the anticodon. Okay, so this anticodon here has to be complementary of the mRNA codon. Okay. So that's how you know like the correct one's in the correct spot. And this is already carrying that correct amino acid. Okay. So then once this moves in, a bond is going to form between these two amino acids. Okay, that's our peptide bond. And then once this bond forms, the whole ribosome is going to shift over. Okay, 3 bases. Okay. So now the A site is empty. Okay. These two have shifted over, and then this one used to be in the E site. It's now going to exit. It did its job. Okay. So kind of think of it as like an assembly line. So the tRNA comes in, it drops off its amino acid, then it's going to leave. Then a new one's going to come in, drop off the amino acid, then it'll leave. So when that ribosome shifts down the mRNA, we just call that translocation. So that tRNA at the A site, it moves to the P site. Okay. That A site is now empty for that new tRNA to come in. And then the tRNA that's in the P site moves to the E site. And then if anything was in the E site, that gets booted and it exits the ribosome. And then finally, the last stage is termination. What does termination mean? Stop. Okay. So this is when we encounter a stop codon. Okay. So when the A site encounters a stop codon, something called a release factor comes in. A release factor looks a lot like a tRNA. It fits the shape, so it fits the slot, but it doesn't carry an amino acid. Okay. So then this is when the chain of amino acids gets broken off and the protein is finished. So the protein can leave the ribosome. So let me show you what this looks like. So at the end, once we encounter a stop codon, something called a release factor comes in. It looks a lot like a tRNA, but do you see how there's no amino acid up here? Okay. So the chain stops, and then the bond here that's holding this together, this gets broken. So then the protein, it's free to leave the ribosome. And so then finally, once that protein leaves the ribosome, it's going to fold up into its unique 3D shape. And then the structure of that protein is going to determine the function. So is it for structure? Is it for movement? Is this a hormone? Is it an antibody? So after that, the protein goes to the Golgi apparatus. Remember, this is like our shipping department. So they process and ship this protein to whatever part of the cell they're needed in, and that's going to depend on what that actual function is. And then the function of the protein, that is just going to make the traits that we studied in our previous unit on genetics. Or even if you think about yesterday, I put up that example of like the white mouse and the brown mouse. So our differences in proteins are going to cause our differences in traits, such as maybe like a white mouse for color versus a dark brown mouse.